Again, just an honor to start off the conservation conversations for 2026. I had never heard of it before, but I always admire Escondido Creek Conservancy's community engagement and really emulate that. So yeah, just an honor to be part of it. So thank you guys for having me. Well, as a native San Diegan and local habitat manager, I've really had the honor and opportunity to explore a lot of our native ecosystems. And in the years that I've been doing this work, I can't help but fall in love with our coastline oaks. So I hope to share with you guys the reasons why I really wanted to pursue research on these trees. But just to get started, I want to give you guys all a little bit of background on some disease ecology. So in the late 1800s, Dr. Robert Koch had first suggested a list of criteria that are used in order to confirm that a particular pathogen is associated with a disease. And so these postulates included first demonstrating that a pathogen was associated with all known cases of a disease, that the pathogen could be isolated and cultured from the diseased individuals, and that the disease could be introduced to healthy individuals demonstrating the same symptoms. And then lastly, that the pathogen could be re-extracted and isolated from the newly infected host. However, we have since learned that satisfying all of these postulates is not always possible, as certain diseases are associated with either a group of pathogens or certain pathogens can't be isolated like viruses. And we have since built upon this concept with the disease triangle. So just to start off here, we will imagine our host being a tree. And this tree lives within an environment under certain abiotic conditions. And in that environment, there might be harmful microbes called pathogens. So the interaction between these three components is what's known as the disease triangle, which is a framework that's used to help us explain and predict complex disease relationships. And while the disease triangle appears to show equilateral sides for these three components, the environment is arguably the most crucial as it can influence the other two. Stressors or disturbances to the environment, such as habitat fragmentation or human-driven climate change, are known to exacerbate disease spread and impact among native ecosystems. For example, studies have shown that drier, warmer winters have allowed for pathogens such as Phytophthora, a soil pathogen, to exist in new areas for longer periods of time. Add this to the increased stress that trees experience with increased drought, and you now have a worldwide epidemic of sudden oak death. So again, this is just an example of how the disease triangle has helped explain this very notorious disease. But now let's imagine a more holistic approach called the health triangle, which looks at how helpful host-associated microbiota influence their host. So the health triangle shown here takes into consideration how the host, environment, and host-associated microbiota all influence the well-being of their host species. Which leads us to the microbiome. And the microbiome consists of a variety of microbiota, such as fungi, viruses, bacteria, and more, that all inhabit different parts of the host organism, such as the roots pictured here. And within the microbiome, we have both beneficial and harmful microbes, which interact with and influence different parts of the host biological functions, like their immune system or metabolism. And while a lot of research has already been done on the plant microbiome in the past couple decades, less is known about the microbiome of seeds. Studies have found that there are two general pathways that microbes get into a host seed. The first is the vertical transmission or maternal effect, in which the mother tree contributes microbes directly to the acorn during development. And so that acorn is already born with a host of microbes that are ideally benefiting its development. The second pathway is horizontal acquisition, in which seed uptakes microbes through external forces, either wind or insects or soil or leaf litter, as exemplified in this image here, where an acorn has fallen on the ground and is now coming into contact with all those microbes. And now that we know a little bit more about disease ecology, I want to emphasize the host species that I'm focusing on for my research, the coast live oak. And as many of you are aware, coast live oak is a foundational tree species that comprises crucial oak woodlands, but it's also known as a keystone plant species due to its ability to facilitate high levels of biodiversity. And this tree supports, Quercus in general, the genus, supports more than 500 species of butterfly and moths worldwide, which is more than any other plant species, which is really beneficial to ecosystems outside of oak woodlands because pollinators are crucial, for anywhere that has a flower or even agricultural areas. And pictured here are just some of the species that rely on these oaks, such as the California sister, whose caterpillars rely exclusively on oak leaves in order to complete their developmental process, and our state mushroom, the California golden chanterelle, who form mutualistic relationships with the oak roots. And for those of us that love birds, I just want to emphasize the biodiversity that oak trees provide to our local and migratory bird species. Oaks host hundreds of local and migratory birds, such as the white-breasted nuthatch and oak titmouse, which many of you probably hear year-round, as well as cedar waxwings and the golden-crowned sparrow, who are visiting right now during winter. And then just to reemphasize the impact importance of coast live oak trees, they provide very significant ecosystem services, not only for the ecosystem, but for us as humans. This includes erosion control with their deep and shallow root systems, the promotion of groundwater infiltration to restore underground water tables, as well as long-term climate change buffers with carbon sequestration and temperature regulations. And true to the name, coast live oak are found all along the coast of the West Coast, ranging from Northern California down to Baja California, as pictured in this map, which displays the two varieties of coast live oak as purple and red dots there. And for the purposes of my research, I am not distinguishing the difference between the two varieties, but I just want to acknowledge that we do have both Quercus agrifolia and Quercus agrifolia occidinea here in California, or in San Diego. And again, coast live oak comprise many significant habitat types throughout the county, from oak woodlands, transitional areas with chaparral, and even riparian forests. And this map here just really exemplifies the full range of coast live oak here in San Diego County. This is a screenshot containing the iNaturalist observations of Quercus agrifolia up to 2024. And you can see how coast live oak comprises a lot of the watersheds in San Diego County, from the mountains all the way to the coast. And these pictures here were taken from various regions of San Diego, which really just exemplifies how mesmerizing the oak canopies can be no matter where they grow. And the biological resource that I really want to emphasize for my research are acorns, which are a vital food resource for many species during the winter and fall when food is scarce. And so pictured here are just some of the animal species that rely on these acorns, including mule deer, acorn woodpecker, appropriately named, and the oak weevil, who actually lay their eggs in developing acorns for their larvae to develop and eventually chew their way out. And be sure to take note of that hole coming out, because that's going to come up later.
That hole is the exit hole?
That's exit hole, yes. And acorns are also a crucial food source for native Californians, at one point making up 50% of their diet. Acorns could be processed into ingredients such as flour, starch, and oils, and are used in very ceremonial and healthy dishes, such as the shawiai dish pictured on the far right there. And it's used, again, for a tool for healing due to its high bioactive compounds. I had the pleasure of trying this dish last year when I visited a couple Kumeyaay tribes, and although it's an acquired taste, it goes really well with some beans or corn. So if you get the chance, try it out. And this leads me to some recent observations that we have been made aware of, a black acorn rot, as we're calling it. And so these black acorns, which are pictured in comparison on the bottom image with a typical unripe, healthy acorn, mature brown acorns, and then on the right side here we have the black acorn, which we are focusing on for the research. And so these acorns were first introduced to me by my professor, Dr. Flores Renteria, when she was made aware of it at a reserve here in Paula area. And they were determined to be non-viable as they kind of had a squishy texture, and when you open them up they were completely rotted. However, there was no sign of herbivory, such as that oak weevil exit hole that we talked about, which leads us to believe that this could be some sort of disease. Without any sign of herbivory, we think this might be caused by an internal factor. And so I've done some literature review, and unfortunately there is no description of such a disease symptom in the literature. And this table here displays all of the known diseases that are attributed to oak species. It comes from the California Oak Disease and Arthropod Database, or CODA. And most of these diseases listed here are either fungal or bacterial. And the only one that's actually attributed to Quercus agrifolia is this drippy acorn disease, pictured on the bottom image there, which is not what we're seeing with these black acorns. The only black acorn symptom that I saw in the literature is pictured on the top right there, which again you can see the reproductive structures of the acorn of the mushroom coming out, but is not what we're seeing in the symptoms described in the research. And so these observations and literature reviews have led me to ask the following questions. What is the spatial distribution of symptomatic coast live oak trees throughout San Diego County? Is this black acorn rot associated with a specific fungal or bacterial species? How does the acorn microbiome differ between these symptomatic black acorns and healthy acorns, or asymptomatic acorns? And is this pathogen transmitted horizontally through the soil to healthy individuals? So my first objective is to determine the spatial distribution of symptomatic coast live oak trees throughout the county. I addressed this objective by performing a morphological assessment of different coast live oak habitat throughout the county. We used a protocol that defined symptomatic trees as those that contain black acorns in their canopy without any sign of herbivory. And again, it's very crucial that we collected them directly from the canopy and not from the ground, because as we learned earlier, once an acorn hits the ground, it's open game for anything, so that black coloration could be attributed to something not genetically passed on. And so, again, I wanted to make sure there was no herbivory, so just emphasizing the photos there. Even though this acorn was discolored and darker, it had signs of an exit hole from a weevil, so we discounted those. And then here are just some images of volunteers that meticulously searching a canopy for these black acorns, because as you can imagine, it's really hard to see a small black acorn on a large tree canopy, but I'll talk a little bit more about the challenges later. And so here's a map displaying the survey sites with letter labels against that iNaturalist map. You can see we covered ground all the way from San Diego City and Mission Trails area all the way north near Fallbrook. And these 12 survey sites were surveyed in coordination with other land managers, such as the Escondido Creek Conservancy. And I focused on areas of denser individuals, but I wanted to access the east, but there was just a lot of, bureaucratic loopholes that I couldn't get through. So in the future, it'd be great if we did this survey again. I'd like to coordinate a survey of the eastern portions. And the goal was to survey at least 10 trees on each site in order to get a good representation, but I'll talk a little bit more about the results from those surveys in upcoming slides. So my second objective was to describe and compare the microbial community assemblage between symptomatic and asymptomatic acorns. And picture here are just a couple examples of some of my acorn samples, where we have healthy acorns on the left side, and then again those black rotted acorns on the right side. And as I mentioned before, you can see the rotted out interior or lysis that was described earlier. And so in order to address the second objective, I'm taking a metagenomic approach with acorn samples collected directly from the tree canopy. As I was going through, I made sure to list the mother tree that I was collecting from for genetic tracking later on in my metagenomic analysis. And so this schematic here just displays the collection plan, where I anticipated collecting from 6 sites. The 3 on the left represent the symptomatic sites, in which I was collecting at least 1 or 2 black acorns from each tree, and then on the right are the asymptomatic sites, or sites that lacked any black acorns. And so I plan to collect acorns from 10 trees of the different developmental stages, because when it comes to diseases, we're not sure if, even though a tree contains a black acorn, there might not be any DNA in that acorn. So it's important to collect healthy acorns on that same tree, as well as unripe ones if you can, because the disease life cycle could be at its peak earlier before the acorn is completely rotted. So just to be safe, we collected a variety. And this gave me a total of 180 acorn samples to process. And so in order to address this metagenomic approach, I used 2 different methods. The first was targeted sequencing, which is represented here on the left, where acorns were grinded down into a fine powder, and then I use a DNA extraction kit to purify the acorn material, and then the DNA samples are sent off to a lab for sequencing specific for fungal and bacterial species. As I mentioned earlier, those are the most prevalent pathogens in oak trees. And then the DNA samples or sequences will be sent back for analysis. The second method is the creation of culture isolates, represented by the photo there on the right, in which we took cross sections of our different acorn samples and then put them in a potato agar dextrose, which will facilitate growth of really anything, specifically like yeast or fungal. And then those organisms are purified into another DNA, which is sent out for molecular analysis. And my third objective is to determine if the pathogen or pathogens from these acorns can be transmitted horizontally through soil inoculation. And in order to address this objective, I set up a greenhouse experiment with 3 experimental groups: symptomatic, asymptomatic, and control, which was just sterile water. And the table here on the right displays the experimental group setup, where there are 10 replicates per group using healthy acorns collected from an asymptomatic site. And each acorn is grown in a separate growth chamber under controlled conditions so that I can monitor them and avoid the potential of any aerial contamination. And so these seedlings were monitored for several months, in which I was watering them evenly and also tracking for any signs of disease cankers. I'm at the end of my research now, but I am collecting data on the allometric traits, such as differences in heights, different leaf production, chlorophyll content, and then I'll be doing DNA extractions to see if there's any presence of the pathogens that we identified from the second objective. And ultimately, I hope that this research can just be used to help inform other land managers about a potential pathogen that's infecting our crucial trees, because we want to make sure that we're preserving our oak woodlands from any sort of threat. And I also hope that my research can be extrapolated to other areas of the Coast Live Oak Range, because as you can see, San Diego is just a small piece of the Coast Live Oak Range, and speaking with other folks throughout my research, no one's really looking at this. So it's important that we keep our eyes on this vital natural fruit. So just getting into some of my preliminary results, as I mentioned earlier, I addressed the morphological assessment to determine the spatial distribution of those symptomatic trees, but it was not without its challenges. As I mentioned, assessing a tall oak canopy for a small black acorn is quite difficult, especially when they're surrounded by poison oak. But you can kind of see that we used a variety of creative methods to get a visual on these acorns. And it's really important not just to be able to see a black acorn, but to handle it. So we tried our best to grab it or throw a rock to get it down. There are a lot of learning lessons. If I were to meet a drone pilot in the future, I would love to have some assistance with that. But that leads us to this table here. And so these are the results of my 12 site assessments. On the left-hand side, we have that same map with the letter labels, which correspond to the different sites there. And each site is represented by a pie chart with 10 slices. The black slices represent the symptomatic trees, shown in black on the column there to the right, and the blue slices represent the asymptomatic trees that were detected on the far right. And so you can see that out of the 12 survey sites, at least 9 of them contained at least one, which was surprising. I wasn't expecting. We had first heard reports of this black acorn in the North County, which has the highest density of symptomatic acorns, but black acorns were found as far south as Mission Trails and like Marion Bear. And again, this is something that, if future research was done, I would recommend doing this assessment again, because I'd be curious if the same quantity and same trees produce the same black acorns. And I also just want to use this time to shout out all the partners that allowed me access, Escondido Creek Conservancy, the Livermore Municipal Water District, the Paula Reserve, and the City of San Diego, who all allowed me access to survey their trees.
So when you do that, you, as much as possible, randomly selected 10 trees?
Yeah,
and that's what you found, 6 out of 4 for Mission Trails? Exactly,
yeah. I made sure, not all sites are created equal. Some sites are 30 acres, whatever their boundary is. Some sites are a swath along a creek. But it was my goal to just kind of randomly pick a tree, and if I could, some trees we spent a lot of time and you just couldn't actually touch the acorns, I couldn't count it. But yes, just trying to randomly survey trees around the site.
That's finding any black acorn on a tree.
Exactly, yeah. And I'm expecting questions later, but just to let you guys know, it really varied. Some trees, and it varied, but probably because of the timing, right? It's such a tight window to survey for acorns, because they just fall right away or get eaten or whatnot. And so I had a very small window to actually, I just did one survey in a day, right? So whether there were more black acorns before I came or after I came, it's hard to say.
And you didn't attempt to characterize the percentage of black acorns on a tree. If a tree had one black acorn and 100 good ones, that was no different than 100 black acorns.
Yeah, we did not make that distinction just because of the time frame it really took to do the assessments. Each survey site took me probably 2 hours, and again, entering the data on the geographic location. And
no, it's not a criticism. It's
just
further work.
Yeah, further work, exactly. It'd be good. Yeah. And then I just wanted to showcase, in doing those morphological assessments, I was really interested in looking at the environmental conditions surrounding each one of those sites to see if there's some sort of correlation with the presence of symptomatic acorns. And so this data, the environmental data that I utilized is from 1981 to 2010, so a little outdated, but I looked at information such as evapotranspiration, soil depth, soil pH, winter precipitation, temperature, all listed there. And then these again are just overview models showing the differences in those different variables across the county. And I used these variables to run a correlation test. And I found that there was a strong correlation between winter and summer precipitation as well as minimum monthly temperature. And so these values seem to be the most correlated with the presence of symptomatic acorns, which isn't surprising because in a paper I've read, they attributed acorn production most to precipitation and temperature. However, I do want to emphasize that this is nothing more than a correlation. That's not proof that these things are responsible for black acorns, because more research, more sampling is needed, and probably more accurate data or more recent data. And I don't want to, sorry, for those who aren't like the most stat familiar, just emphasizing that the p-value is really low, which means that is why that variable has that strong correlation or curve with the independent variable of symptomatic acorns. And then on to the second objective, in which I'm trying to determine or describe the microbiome. We have prepared all the acorn samples as much as I could. So I mentioned earlier I was trying to, I calculated that I would collect 180 acorns. I actually ended up collecting over 300 acorns because on each site, I realized I was finding more black acorns than I expected. And so I couldn't help but pass up the opportunity to collect them. And so my original plan turned from doing 6 sites to all 12 sites that I surveyed. And so it took a long time to do the DNA extractions. I hit a lot of obstacles along the way. It's actually a lot more difficult than you think to extract DNA from an acorn versus leaf or root matter. And so I was hitting hurdles with just getting viable samples. But I'm at a point where I've sent out some samples and I'm waiting to get results back from a lab that is able to process them. But I did do some preliminary targeted sequencing in 2025 based on some samples from 2023. And when I sent those samples out, they were positive for some fungal species listed here. The main one I want to highlight is Pneumoniopsis pericobulata. I'm just saying that confidently, I don't know if that's how you really say it. But that species is a relative to another species in the same genus, Pneumoniopsis, that is attributed to a disease called chestnut rot, found in China. And it's really interesting to note because chestnuts and oaks are in the same family, the beech family. And so again, this is just kind of leading me down the path of wanting to explore this individual more. But there's no literature on this particular pathogen. I don't know if it's local. I don't know if it's been introduced from agriculture. Some of these are from agriculture, like Altanera tenuissima is usually found in winery crops. So I'm imagining that a lot of this has somehow made its way from nearby agricultural areas. And then secondly, I've had some preliminary information from the culture isolates. We sent off some samples already and again identified another suspicious microbe, Peroniliae, or I'll say it that way. That one is a suspected necrotrophic fungi, which means that it breaks down the plant material and eats it. And it's commonly found in pea plants, pictured here. So you can kind of see the root rot that's been caused by that fungal species. And so again, that was found from the culture isolates, which again leads me to suspect it as being maybe one of a complex of things that might be affecting these acorns. And so not much information is known on it, but again, as I look at more of my samples, I plan to assess if this is one of the higher present species. And that just leads me to my acknowledgements. I just really want to thank USDA for helping to fund this project, as well as the Bloom Hayes Ecological Research Grant. And again, thank you to all the partners listed below that granted me site access, as well as just helping to survey with me. A big thanks to Dr. Lipson and Flores Renteria for their continued guidance, and all of my lab mates who gave me training. As I mentioned, I'm a habitat manager, so I'm an ecologist. And there was a lot of evolutionary biology involved with this, so I was learning a lot of new skills along the way. How to do DNA extractions, how to run a PCR. That was all new to me, and I hope that I was able to do them justice, in speaking to you guys today. But yeah, with that, I am open to any questions. So again, just thank you all for your time. There's some questions during, but
from your culture there, so you think it may be multiple diseases causing this?
Yeah, and that's something I alluded to in the beginning, but usually a disease is not the result of one single microbe, it's a complex of things. And so that's why I'm curious to find out. I don't have any actual figures that I've created just yet, but usually you'll find that there's A, B, and C linked with this particular disease. That's what I'm curious to find out, is if these black acorns all contain similar groups of microbes.
Excuse me if you mentioned it before we walked in, but did you notice any difference in the appearance of the trees themselves?
That was one thing that we were assessing as canopy health, and I was really referring to the U.S. Forest Service's gradient, 1 through 5. 5 being the worst, it's almost barren, 1 being completely fine, and there was no apparent trend. That you'd find a fairly healthy looking canopy, or some with some defoliation and no black acorns, right? So, that was one thing. There was no signs of exuding sap. I'm really aware of other pests like gold spotted oak borer or shot hole borer, and so that's kind of one thing we're looking for too. Is this a secondary infection? Because oftentimes when a plant is under stress, it's easier for it to exemplify or display some other symptom. But at this moment, no. We didn't. Unfortunately, I didn't have the time to quantify how the ratio of symptomatic to asymptomatic on a tree, and also didn't see any other symptoms. Yeah.
So what is the solution, right? If it's in the soil, like, does that mean that every new tree is gonna like end up with it as well, or a high likelihood, or is it, if it's from a host, right? Like, if we're trying to transplant or put new oaks in an area, what would be like a way to prevent, bringing in a diseased oak?
No, absolutely. I think best management practices are always a crucial component to restoration. Whether you're doing silviculture, right? You don't want to introduce some sort of diseased individual. And I would say, as of now, that is the practice that I would implement in my preserves. I have my my replicates that I'm studying, and I don't plan to plant them anywhere because if they are diseased, I'm spreading something. But in the long term, more soil analyses would be appropriate. You can see I didn't have the to include that in my research, but that would be a secondary research item to do. And if so, what are any soil amendments that could be made, or what are the sources of that? So yeah, best management practices, I would say, are always safe than sorry. But it's really the main, I would say, interest from this research was the polyp reserve, because they collect acorns pretty regularly, and they they want to make sure they're not ingesting any harmful microbes. And so for the time being, they are holding off on those regular ceremonial purposes. But yeah, just hoping to learn more during this research. But
From all acorns, or just from acorns from trees which you said are black?
just from agrifolia, we have not observed this in any other species. Anglomanii seems to be fine. Demosa seems to be fine. Bearbrifolia, but just Quercus agrifolia for the time being. I'll go check.
I stepped out a couple times, sorry if you covered this. Is a mutation a possible explanation for this?
That's an interesting thought. I mean, I'd have to defer to my evolutionary biology professor to ask her if that is a feasible explanation, but...
Mutation of what?
Well,
Tree or
it
the
seems that
pathogens, or-
it's happening on only some acorns of one mother tree.
Right.
So if it was a disease that the tree had, you would expect, all of them to have them, or the majority of them to have them. So I was thinking, maybe it's just some little mutation that's kicking off, that's causing these symptoms.
Yeah, that is, I mean, I think that's a really good question that I would hope somebody else could explore. Yeah.
It's not gonna be me.
Yeah. Yeah, no, early on, I was, someone pointed out, that picture I had where there was like a black, healthy, and unripe acorn altogether. They're noting like oak trees, which I learned have like sanctioned root systems, so if like one part of it gets diseased, it could just kind of lose that limb.
Yeah.
And so that's why they kind of mentioned what you said, right? Like, wouldn't you expect all of them to be black? Which is kind of like going against the hypothesis that it's vertically transmitted, right? So that probably seems like another valid explanation. So I hadn't pondered that before. And sorry, there was another question, yeah.
Did you interview native elders to find out if there was any historical knowledge about this disease?
Oh no, I had not. I was mostly corresponding with the Polyp Reserve contact. They have, I don't want to say they have more exclusive rights to speak with me or anything, but I would like to pursue that. I had not, but yeah. As far as I'm aware, this is a new observation to them, which is why they kind of raised their hand about it. So I'm not sure. The black acorns, yeah. It's a fairly new observation, and that's why I was made aware of it. And like I said, I manage preserves in the Escondido area, and I hadn't even been looking at the acorns until my professor mentioned it. So who knows how long it has been going on, but as far as I know, it's new to the area. Yeah.
So it's an introduced species?
Or mutation.
Or
Anderson theory.
Yeah, a couple things I was considering is it's either the result of a culmination of just poor environmental conditions, that's now expressing itself, or it is a disease that is, maybe in tangential, just being more expressed as well. Hard to say at this time. Yeah.
When you did see a tree with black acorns, like just your general sense, how many acorns does the tree tend to have? Is it all over the place, like from one to a whole bunch?
It varied quite a bit depending on the size of the tree. You know, a lot of the trees that we surveyed are, you know, were in the 20 to 30 foot range because it was the easiest to see. So a little bit of survey bias there, because when I was here at the Elfin Forest, it was really hard to see a lot of that taller canopies. But I would say it was probably on the lower end, you know, somewhere less than a dozen were black. Whereas I went to some sites, I went to one tree in particular in Mission Valley, or Mission Trails that I recall, that was, you know, probably had 30% black acorns. And so, but that was out of the norm, you know, they were usually on the lower end.
It didn't make a difference. Your map showed that there were some groves that were 100% of the trees exhibited.
Right, they at least had, well that,
At least one.
At
yeah,
least one,
yeah, exactly. The pie charts are just for trees, so,
Right.
yeah. So Sardina had 100%, I think.
Yeah.
Yes.
We
Yeah.
noticed that.
Yeah,
Notice that,
yeah, like it was not hard to find a tree with black acorns at that site. Yeah.
If you do get in contact with a drone person, you might be, have you talked about remote sensing for data collection? Because that unique color may be something that remote sensing can
Absolutely.
pick up like that, which would make your data a lot better.
Yeah, that's a learning lesson going along, just be like, oh yeah, I can't just like see with my binoculars. It's like shadows or whatnot. But yeah, we were thinking the color signature could be trained in a drone. So yes, if I had more time, I would like to do that.
So this is not gonna be a PhD topic, right?
Yeah.
So you were talking about like the different variables, and you said precipitation like mediates or moderates it. Like how does that happen? Like do you guys know how like precipitation affects like or creates more of the black acorns or?
Yeah, no, I at this moment that's as far as I am, to be honest. You know, from the literature I've read, the lack of precipitation is what stresses the tree and also facilitates the longevity of that soil microbe. You know, they kind of like do their life cycle while it's dry, you know, and it gives them longer time to do that. So they're just kind of expressing themselves more.
Which direction was the correlation on minimum monthly temperature? So high minimum monthly or low?
The high minimum, the
So
higher
the
temperature.
warmer we are.
Yeah, the higher the
more-
temperature.
the higher
So kind of
correlation.
alluding to this climate change, and my professor would scold me right now, I should have emphasized that again. Yeah, increasing climate change conditions that we're seeing will eventually, potentially lead to a higher increase of those symptomatic, yeah.
You said it's when these black acorns start to form. I know you had limited time to study, but
Yeah, that's one thing I'd also love to do is maybe to put some sort of like camera, just like you don't know which acorns are going to be black, right? But it's like, does it start, because some acorns I'd see were small and black, you know, and some of them were some were partially black, right? Some were partially black. So I never really got a chance to see that development, right? Like was it mature and then just turn black, or does it just go from green to black? I really just, it just opens Pandora's box and more questions, right? Yeah.
What are you trying to do with your seedlings as far as studying them?
Oh, well, like I said, so if I had more time, I would grow them out for years and years and years.
See if they make these black acorns?
Because, yes, exactly, but it takes like 7 years or so till they like can produce their acorns of their own, so not enough time for a master's.
world's longest master's program.
But as of now, I'm just looking at, like I said, those comparable allometric properties, you know, just things I can measure without harming the tree, the sapling so much, right? And then doing DNA extractions to see if there's any presence of them.
Yeah, see if it's there.
See if it's in the tissue, you know.
Okay.
So if you find it in a black acorn, you find something in the DNA, you can test your seedlings and see if it's there.
Exactly, because one thing I've read is that pathogens will exist in just the epidermis of plants, you know, and then it's passed on to the seedling during that developmental process. But yeah,
Rewind on the title of your dissertation,
determining the presence of a potential novel pathogen in coast live oak.
okay.
Yeah.
So, and the novel thing,
Is a new disease because it hasn't been described in literature. Because we know what,
I mean, black acorns have been around though, right? I mean,
not that we're aware of in coast live oak.
in coast live oak. Oh,
Yeah, I've looked at a lot of research and it's never been described.
That way.
Yeah.
Okay.
There's a lot of, and again, it'd be cool to use maybe some sort of device that could like really tell me, is this what what hue of black this is, because sometimes the acorns are so brown, just super brown,
Yes.
you know, and you know, there's a lot of morphological variability in acorns, but the ones that were black just really stood out. But
And they have different structure, you said.
yeah, a bit of a squishy texture, which indicated some sort of lysis inside of the, you know, deterioration of the embryo. So yeah. And
I caught something you said that there were some acorns that did not have DNA.
the viability of the DNA was hard to explain.
Oh, the viability of the DNA.
Yeah, so when
After
you're
they tried to extract it, it didn't. look
yeah,
like
there's
it.
not. So the kit we were using, it's the Pathogen plant mini kit, and it's like I said, normally used for leaf or root or stem material. But when you're breaking down an acorn that you've frozen, you have to use liquid nitrogen, and so potentially it could be breaking down. Like that process could be breaking down the DNA entirely, and so it's a fine line between getting the DNA to a point where you can liquefy it versus completely masticating it and not having anything. But you won't find out for 5+ hours because you have to like, you know, go through the process, put it in the electrophoresis gel, and see what comes up. And sometimes you're looking at a blank screen, and you're like, what did I do wrong? Yeah, so. Yeah, so DNA viability was one of the bigger challenges that I wasn't anticipating. Yeah.
Have other Indian reservations or tribes expressed interest or shared the problem, or what about like Santa Isabel and those?
Rincon is one that did actually reach out to me. Unfortunately, I was kind of already like past that phase. I did all these assessments in November of 2024 just because I had such a small window. But they reached out to me kind of near the end of that season, and I was already past my ability. But I'm hoping, like my goal is that from learning from my current collection, my goal is to come up with some sort of quick cheat sheet, right? Like I don't want every land manager to have to go through what I went through to see like, is this the result, right? So I'm hoping I can create like a quick white pages type of, this is the symptom you should be looking for, here are the characteristics, whatever it is, the texture of the acorn, size of the acorn, time of year. So I'm hoping that it can be applied to other, I can maybe just go back to those other interested groups and more or less have a better idea just from what I'm doing. So yeah, people are interested, I just can't say I was able to work with them this time around. Yeah. Saw some other hands. Yeah, no, yeah, it's been really exciting to just, like I said, explore different parts of the county, work with different community members. You know, there's a lot of us that care about oak trees, and I don't know if you guys know Jim Crouch at all, but he's an amazing guy that just reached out to me about planting native oaks. So if you guys are interested, he's got over 6,000 plants that he's looking to get rid of. Yeah, so, and as far as I know, he doesn't have any black ones. But you know, I've kind of shared my research with him a little bit, and he's going to be replanting some of the areas where we've lost a couple trees here in Escondido. But yeah.
Oh, I got another question.
Yeah.
If in our hikes we come across a black acorn, can we let you know somehow? Or?
Yeah, please do. First off, I always suggest mapping things on iNaturalist. If you use that app, iNaturalist.
Yes.
And you can make a note. You know, make a comment in there and put a picture specifically of that black acorn. That way, either
On the tree.
yeah, on the trail. That way, I can save me time and I can look at the picture on the computer. But yeah, please feel free to reach out to me. Yeah.
Okay.
Yeah.
That might be another outcome you could do on this if you've got interested parties, is try to get some citizen science going on collecting a broader incident report.
Especially if we expand to northern
Right.
county.
Or
Yeah.
up and down, even in Mexico.
Yeah, exactly.
Because nobody's reporting this and starting here, or maybe nobody's seen it or thought to talk about it. But if your report gets out there, it should know something they can do.
Yeah, that will be definitely something I include in my recommendations.
I know this is for your master's, but do you plan on still pursuing this research as a habitat manager, or do you plan on handing it off to a future student?
I guess it depends on who comes into the lab in the next 5 months, but at the same time, you know, working as a land manager, I do plan to continue this in some capacity. I know I won't have the same access to labs, but I absolutely plan to continue my morphological assessments and discussions with other land managers. You know, I'm part of the GCCO, which is a global group of oak conservationists, and just hoping that in that continued relationship we can learn if other entities observe these in other parts of the world. So yeah, the lab work is not my forte or passion, but I've learned a lot and appreciate a lot for people that do work in the lab. So maybe I can help train someone to help me analyze acorns going forward.
What land do you manage?
So I'm with the San Diego Habitat Conservancy, which is all around the county. We're a pretty niche nonprofit that kind of works in the mitigation realm. So we have sites here in Escondido. You can go to our website and see those specifically, but we have a couple in Bonsall, down south in Otay, even as far east as McCain Valley. So we have quite a few different biomes, but I would say oak trees comprise a lot, or are found on a lot of our preserve areas, which is how I had a lot of access to sites too. So, yeah.
Thank you.
Thank you, man.
Great. Thank you for your questions, guys. Really good discussion.
Rather than find your way out if you're so tired. Thank you, everyone.
Oh, good. All right. I think you see.